Welcome to the next series, the next video in oncology for non-oncologists. Myself, Dr. Gautam. I am a medical oncologist from Hyderabad. So, another important hallmark of cancer is growth promoting metabolic alterations. So, if you see, cancer cells are like normal cells need nutrients to survive. But as told, the human body can serve need of every organ, but it can't serve greed of every organ. Cancer cells are greedy. They require more amount of nutrition than normal cells. So to complement or to acquire more number of nutrients, they need certain modifications of the normal metabolic machinery so that they can survive in this nutrient stressed environment and adapt themselves to the nutrient stress environment. So the most important metabolic alteration that we see in cancer patients is aerobic glycolysis called Warburg effect. It was invented by Warburg, so it is called Warburg effect. So what happens? What's the difference? Cancer cells, every tissue in our organ has a specified function, but the only function of cancer cells is proliferation. So what do you need for proliferation? What you need for proliferation is complex carbohydrate molecules which form the skeleton of a different cell. Where do we get this complex carbohydrate? Normal cells or any cell will get the complex from the proteins or nutrients intake. So for the cells to have proliferation, it is necessary for a lot of complex carbohydrate molecules. So usually in with this scenario, let us get detail into Warburg effect. Even in presence of ample oxygen, cancer cells demonstrate a distinctive cellular metabolism called glycolysis. So usually we all our cells will undergo Krebs cycles in presence of oxygen, glycolysis in absence of oxygen. But cancer cells, even though in presence of oxygens, will go for glycolysis. Why glycolysis is bad? Because like glycolysis uses lot of energy, lot of uses will supply the cell with less number of ATPs when compared to Krebs cycles. So at what compromise? Why is the cancer cell compromising the ATP production capability of a normal cell just to glycolysis? That is something. So what does aerobic glycolysis do? Aerobic glycolysis is glycolysis happening in presence of oxygen. What does it do? Aerobic glycolysis provides rapidly dividing tumor cells with metabolic intermediates that are needed for cellular synthesis component. So it serves the function of the cancer cell, which is function of cancer cells is only proliferation. This phenomena is called Warburg effect. This shift of metabolism is done through something called metabolic reprogramming. So through metabolic reprogramming, cells, instead of undergoing cancer cells, instead of undergoing Krebs cycles, undergo aerobic glycolysis. This reprogramming is called Warburg effect. Why is Warburg effect important? Which is specially applied in case of PET scan, where FDG glucose molecules are more concentrated. FDG is a radioactive isotope that is used in PET scan, is more concentrated in the cancer cells. Thus, we can differentiate between which cell is cancer, which cell is not cancer to most of the extent by using FDG PET scan. So this is a fraction. What does the thing? So usually glucose is split into different carbon molecules, and the energy that is bind between carbon molecules is converted into ATP. But instead of glucose shifting into simple carbon dioxide and water, it is now converted into pyruvate and lactate, thus producing only few amount of ATP. This thing is what happens with the help of glycolysis. First thing, it produces rapid ATP synthesis. Krebs cycle is small. Warburg effect is fast. It produces rapid ATP synthesis. Second thing, it helps in the tumor microenvironment. The thing is that it enhances the disruption of the tissue architecture and immune cell invasion. Third is it is responsible for signaling and biosynthesis. It as told, it gives a framework. It gives intermediates for the synthesis of several other things. So this is a quiescent cell. This is a normal quiescent cell. This is a growing cells. So usually Krebs cycle happens, but in case of a cancer cell or growing cells, what happens? Instead of Krebs cycle, the increased glycolytic intermediates are used for protein synthesis, nucleotide synthesis, and lipogenesis, which will prosper. This is the difference between normal cells and abnormal cells. What are the different pathways of metabolic reprogramming? They are receptor tyrosine kinase, PIK3 kinase pathway, MYC protein expression, antagonism of STK11, production of oncometabolites, and avoiding autophagy. The important pathway, PIK3 AKT kinase, what does it do? It upregulates activity of glucose transporters and multiple glycolytic enzymes, thus promoting aerobic glycolysis. In addition, they also phosphorylate and inhibit pyruvate kinase, thus providing more and more substrates for aerobic glycolysis in cancer cells. These intermediates are then siphoned off for the synthesis of DNA, RNA, and protein. So PIK3 kinase, activation of PIK3 kinase will produce activation of AKT, which causes metabolic changes. These metabolic changes will cause growth, survival, growth, and protein. MYC gene is master transcriptor of the cell. So there will be no cancer cell. Most of the cancer cells will be having MYC gene. If not initially present, ultimately the MYC gene mutated. So what does it do? It upregulates multiple glycolytic enzymes and glutaminase, which is required for mitochondrial utilization of glutamine and other intermediate sources of other important sources of cellular components. So STK gene is a tumor suppressor gene responsible for Peutz-Jeghers syndrome. It antagonizes metabolic changes. Intact STK gene antagonizes Warburg effect, but mutations in STK gene will the antagonism is lost, and hence it will promote Warburg's effect. So mutation in STK gene leads to lack of inhibitory effect, promoting Warburg effect and oncometabolism. IDH gene mutation is usually seen in glioblastomas, and IDH gene mutation it is an enzyme. What is IDH gene? It is an enzyme that catalyzes oxidative discarbylation of isocitrate producing alpha ketoglutarate. So formation of IDH mutation will lead to production of 2-alpha hydroxybutyrate instead of alpha ketoglutarate. This 2-alpha hydroxybutyrate acts as an oncometabolite, and this oncometabolite, what does it do? It acts as an inhibition of TET2. And what does TET2 do? TET2 is it will enhance the DNA methylation. And abnormal DNA methylation will leads to misexpression of cancer genes. These cancer genes lead to transformation of oncogenes. So how did it start? Usually IDH will produce alpha ketoglutarate. Mutated IDH will produce hydroxy alpha ketoglutarate. Hydroxy alpha ketoglutarate will inhibit TE2. So TET in its actual form acts as a tumor suppressor gene. The inhibition of inhibitor of TE2 will lose the tumor suppressor proteins. Loss of tumor protein will lead to abnormal DNA methylation. The abnormal DNA methylation will drive the genes in cellular transformation on oncogene depicted in these things. So 2-alpha hydroxy metabolites will cause epigenome regulatory enzymes, which cause epigenetics changes, leading to altered gene expression and ultimately cellular, and as expressed here. So another important part is once a cancer or the nutrients, once signals are there, usually these cancer cells undergo autophagy. But cancer cells escape this autophagy. How do they escape this autophagy? Is by altering the reprogramming its energy metabolism. Normal cells, when nutrient depleted, go to a state called autophagy, in which cells do not only arrest their growth but also cannibalize their organelles. Tumor cells also grow in this nutrient environment but do not undergo apoptosis, because tumor cells will disrupt the pathways of apoptosis. There are several genes that promote autophagy, which acts like the tumor suppressor genes. Means lot of autophagy also promotes cell growth. Thank you very much. End of this slide.